Be Jun Wang from BGI. I must admit that there are very few occasions that we have the pleasure to have in the same session two of the most prominent figures in next generation sequencing of our days. And actually very few meetings that we have this experience. So I'm not going to talk about Jun because it's going to take me actually longer than your talk. He's professor of bioinformatics in the University of Beijing. He also has an adjunct professorship in Copenhagen. He has more than 200 papers in top quality journals like Cell, Nature, and the like, New England Journal of Medicine, received several prizes, and his research interest focus on bioinformatics and whole genome sequencing to elucidate the genetic base of complex diseases. Jun, thanks for coming. We really appreciate your coming here, especially with your busy schedule, and we look forward to your talk.
Thank you, George. Thank you very much for inviting me to this meeting. It's actually very nice to talk after Eddie because he actually set already the technical basis of my talk. So I'm really going to talk about one project that BGI will focus on the next 5 years about the Million Genomes project. I'm just trying to introduce the rationale why we should sequence the Million Genome and how those Million Genomes sort of compose of. Biology has become information, right? So instead of just generate hypotheses and test true sort of approach, it become a more information and data-driven thing that people don't really think about hypotheses before they're doing research, but they start to generate a large body of data, then construct hypotheses afterwards. So this kind of data-driven approach become more and more dominant model for this century of biology. As you can know, this sort of branded man touching the elephant, the only way really you can paint the elephant is you get the whole picture of elephant. Sometimes actually when you're doing very, very difficult analysis in the bioinformatics way, just because probably you don't really have complete data or enough data. Why we need a million? Well, the history back to the Human Genome Project, why we actually need the first Human Genome Project. Right? At that time, we have EST cloning, we have PCR technology, we have many of those sort of one gene started tools, but why we actually need one complete human genome sequenced. Because we actually need a whole picture of a human genome. We don't really set up any hypotheses of how many genes of this human genome, what's the GC content, how many repeats there, and what's the real sequence of that. You should just read it. BGI was like part of the human genome from the very beginning. We did 1% of human genome as sort of one of the 6 countries contributed into this project. But after that, people are really wondering, is one genome enough? What's the next step? People were talking about post-genomics era, right? So genome is already finished. Now it's functional genomics. But actually it's never finished, right? So the post-genomics era is really not really sort of coming in a way because we will stay in a genomics era for a very, very long time. We started first of all, like 5-6 years ago, we probably published the first Asian genome in Nature. I like this quote very much. They say, your life in your hands. At that time, a personal genome means you sequence many of those short reads. They actually align back to the reference genome. You get lots of SNPs, normally 3 million SNPs. And many of them are novel SNPs you haven't discovered before. For example, here in this Asian genome, it's like 500,000 SNPs hasn't been really discovered before. That comes into a very nice Nature cover. But we know actually the reference, mapping the reference is not enough because you actually missed out all the online region, right? So if you have a very complete, sort of complicated region as structure variation, or you actually have a novel sequence people don't really sequenced before in the reference, how you can deal with that. So for this first Asian, we discovered, for example, 5 mega base pair novel sequence, which cannot be seen in the reference, which was mainly a Caucasian. Then we actually started looking at those novel sequence. They have lots of putative genes there. It's about 100 putative genes, 1,000 putative genes. And more than 100 has a RefSeq database backup. That means they have RNA sequence aligned to that. In other words, probably functional. So those things have become very interesting, and we start to sampling 41 populations across the world, about 400 samples, about those novel sequence. And we discovered this very interesting population pattern there. So it seems like every population have certain private sequence, or every even individual has certain private genomes, which are not just copy number variations or single nucleotide substitution there. They're actually yes or no variation there. So well, eventually, by just mapping technology or exomes or anything is not really enough. As Reddy already pointed out, you actually need a de novo assembly, which we done for this Chinese. So you need to really start from the scratch. There is really no reference. You just have to sequence the individual completely de novo wise. But anyway, you can get a lot from just one genome, right? You can study the human history, for example. We've done 4,000 years ago a Greenlander sequencer. We actually did Aboriginal Australia. We started another Mami whatever genome project in China, which we have access to over 1,000 of Asian DNA. Some of them, more than 5,000, 6,000 years ago, and some of them even, a little bit older there. The quality of DNA is really a challenge, but actually you can really get a lot from just one genome, the one Asian genome that's sequenced. But we know it's not enough, for example, to study the medical genomics, right? It's really your life in your hands there, even you sequenced one genome there. I mean, I asked the first Asian we've sequenced, he is the chairman of VGI, Dr. Yang. He got his genome sequenced. I sort of asked, is there anything you feel like you want to change after you sequence your genome? The answer is no. I mean, there is actually, it's not, you don't really know a lot by your genome, especially when you are healthy 50 years individual. Then it comes to what's really ideal for medical study there. When we talk about ideal analysis, it comes to really, you need to have a very full sort of coverage of clinical data. You need to have whole genome. And then you need to sequence many, many of genomes there. So this is really true. So we have some of the, I mean, if you look at all these different dots here, so people initiate lots of projects which are just trying to find a shortcut. For example, 1,000 Genomes Project is studying all the population genetics without any phenotype information. Or sometimes people focus on one gene, but with phenotype information, but only one gene. Or sometimes people get a set of genes based on hypotheses and started to sequence many samples there. And sometimes people just expect to get a one genome sequence with some phenotype, want to give some sort of ideas about how to do intervention. But eventually, I mean, those projects are not enough. I mean, for example, the 1,000 Genomes Project, BGI is part of it, and we've sort of done the Asian part. We discovered, again, many, many of those rare variations there. And if you look at all the rare variants, again, those variants are very population-specific and individual-specific. So it's about more than half of them you can discover. Is that too bright, actually, for this? But anyway. Sometimes if you look at the population-specific genotype-phenotype associations, it gives you very interesting results. This is the one of the sort of results we published in Science. So we discovered Tibetan Chinese. Those Tibetans are really high altitude adaptation. And Han Chinese are, close sort of nearby population, but cannot really climb mountain, very high mountains there. So we sequenced the Tibetans, and we also sequenced the Han Chinese. Then we discovered this one gene, for example, the EPAS1 genes there. If you look at the Tibetan population, it's over 90% of them are very, sort of having these mutations there. But on the Han Chinese, it's less than 5% or 8%. So it becomes very interesting. If you look at these one genes there, they are positive, highly positive selected, and have a very, very strong association with blood hemoglobin level, which we know is needed for when you actually climb a mountain there. So this is it's really true. So if you look at the population-specific SNPs there and started to think about maybe there is a phenotype associated with that, that would be very interesting. Of course, it's not always like that case. You will find out genetic drifting a lot, right? Sometimes you find out actually population SNPs without any phenotype implications. But this is just one example to tell you how to make those links there. For the disease genomics, if you look at any kind of complex disorders, they have a very wide disease allele-frequency distribution there. And people will really focus on the common variation by GWAS analysis. It has been really successfully happened. I mean, if you look at all the chromosomes, people, I mean, this is from the NIH database. you can see, I mean, almost every chromosome are full crowd of all those kind of disease-related genes there. But unfortunately, there is certain disappointment there. If you look at type diabetes, for example, there is about close to 40 risk loci associated with type diabetes there. By GWAS analysis, very highly sort of frequent SNPs there. But the biggest odds ratio for this TCF7L2, which was actually the first gene was discovered by GWAS analysis, the odds ratio is about 1.3 or something like that. So add them up. For the entire type diabetes, only expands something like 6% of the heritability. There is a big part of the heritability has been missing. So they've been sort of quoted as the dark matter. How to really find the missing heritability become a big issue there. And we started the first attempt trying to sequence many of exons of individuals there. So this is actually the Sino-Danish analysis. So we collaborate with the Danish scientists. We actually sequenced more than 1,000 cases and more than 1,000 controls there by whole exome sequencing. We got published in the journal here, but actually there's a 3-stage there. The first stage we sequenced 2,000 exomes there, and find out the interesting SNPs started to do genotyping association on the 17,000 SNPs in say 16,000 Danish individuals there. Then we actually narrowed down to very limited number of SNPs there and started to do replication on 63,000 European individuals there. The results comes to actually discover, for example, here, 3 interesting SNPs there. Not really much. It's kind of disappointed a little bit. The reason could be simple as we've only done 2,000 exome sequencing. It's not enough, right? We need to do 20,000 individuals, or 200,000 individuals. Maybe gives you different results, but we don't really have the technology yet. That's also part of the reason we sort of acquired complete genomics, because for the next step there. But anyway, so before we can really move to a rare variation study there, I'm going to tell you one of these extreme rare variation that we are really working on is so-called de novo mutations there. So we initiated this project with Autism Speaks and all the other partners there to study an autism genome of 10K projects, right? So the 10K Autism Genomes Project is focusing on trails and de novo mutations, and also all these kind of recessive genes and so on. We first published this paper in Cell last year as a cover story there to sequence 10 identical twins with autism cases and normal parents there. And we discovered about 600 de novo sites, which is quite a lot. One of the things there is very important technically it's important to identify the de novo mutations, because sometimes a sequencing error or anything, amplification error or anything like that could show up as a de novo mutation. So you need to develop a very sort of precise tools to make sure the mutation you find out is real, technically and biologically real. The good thing is that we've been sort of developed this technology, which can really make like over 95% of the sensitivity and specificity for the de novo mutations there. Then for the de novo mutations, we know they play a very important role there. They have lots of different factors to affect the de novo mutation, like intrinsic intrinsic factors and extrinsic factors, like parental ages there. So I'm just trying to show you some of the, for example, this is the parental ages there. So as we can see, sort of 1 year older, if the parents, for example, the father is 1 year older, the child have 1 more de novo mutations there. So it's very clear trend. It's like 1 year, 1 more de novo mutation there. Actually, if you look at all this distribution of the genome, those de novo mutations are not randomly distributed in the genome. So if it's random, it should be, looks like that linear. But it's actually really non-random here. This is the observed curve there. And they actually fall into the chromosomes like this pattern. So they are clustered together. Sometimes you find out very warm spots, like many de novo mutations. Sometimes you find out very small and very extremely high dense region on the de novo mutations. If you zoom into these regions there, the first thing we've sort of found out is very interesting, because it's a lot of de novo mutation. We thought if you compare cross-species comparison, they will be less conserved. But the results is actually they are more conserved. It's kind of interesting. So you discover this sort of pattern, not just from gene level, but from exon level too, both cases and controls there. So if you zoom into those regions there, of course people are talking about different sort of hypotheses, like if those regions cross species are very conserved, it means they are actually functional. They are really, really useful from evolutionary perspective. So they are actually, even they have lots of de novo mutations, they are adaptive mutations there. Or they're still under purifying selection, or they probably couple with DNA transcription or DNA repair. So in any of those cases, they're just trying to say those de novo mutations are really, really functional. So then we have to really sort of zoom into the regions that are clustered with de novo mutation. Then we find out the disease genes, for example, from all this OMM database there are more sort of centralized into those regions there. And if you look at the gene catalog here, there are actually many of those kind of brain development genes and autism or autistic related genes there. So again, it becomes very, very interesting whole story. As you know, the average generation, so the baby generates something like 60 to 100 de novo mutations compared to the parents. So they are clustered in the genome. If you look at those regions there, they are full of those disease genes there. If you look at those disease genes there, they are actually enriched by those brain development genes. So this is actually basically the whole sort of complete story there, which is kind of very interesting. Then you understand why the father's age is older, the kids to have autisms, the risk is higher. I start to worry myself anyway. So we started to screen all these families. For example, this is the ASD families with all the genetic variations there, as you can see, which is quite a lot. So then we sort of try to use de novo mutations, X-linked, and also recessive genes model, trying to understand why. I'm just give you some example. For example, here, de novo mutation in these genes, which is really similar to FMR1 genes there, which has, Fragile X and so on. Gives you know exactly about these de novo mutations. This is another de novo deletions, which is a really 1.7K deletions, which have in one of the families there. So you can really start to analyze many of those families and understand why they have autism there. So I explained the extreme rare mutations and also explained a little bit part of the common variations there. But to fill in this part, fill in the complete picture of any of those complex disorders, you really need a meaning whole genome sequence with complete phenotype information. Then it will make you sort of your health in your hands there. As already said, we actually need the platform to do that. I'm not going to repeat his talk. Now, Complete Genomics and BGI is just one big company. And if you look at actually the Million Genomes, for example, at the individual level, so I'm just trying to tell you 3 applications that we are really focused on develop the Million Genomes concept. The first one will be how to have a healthy baby, how to really sort of reduce the birth defects. In China, the birth defects is about 5.6%. And we have this one-child policy, so it's really a big deal in China. So we developed this non-invasive prenatal testing there, so we can actually, from maternal blood, we can basically test a complete genome of the baby, right? So then you can really start to do lots of testing there. So we've done already about 200,000 samples in China. We discovered more than 2,000 T21 Down syndrome kits, which is really effective. And more than that, we are starting to do all this kind of offering genetic testing for over 600 different diseases there for any of those newborn babies there. This is the first part, right? To have, well, before that, you need to even screen sort of pregnant, to screen the couples, the genetic carrier. So if they actually carry a kind of genetic recessive gene disorders there. But anyway, so this is just one part to understand how to have a healthy baby. The second part would be for the cancers, right? So we've started the single-cell genomic sequencing. We published 2 cell papers last year about, you can pick up 1 cell of a cancer tissue and normal tissue and sequence its entire genome there. The idea is simple, right? Because we know for final chemotherapy-resistant recurrency cells there. For any of those cells, you can always trace back to the fertilized egg. And how to understand the cell lineage tree along this, and how to understand what kind of environment factors and what kind of driver genes is making all those somatic mutations is very important there. Just give you one example on the bladder cancers we've done. So as you can see, this is just one cell, each row, and this is different genes. You can after sequence of each cell, you can clearly have the cell lineage tree here. From there, you can infer back the history of tumor progression. You know exactly when, what kind of gene had a mutation become the separation between tumor and normal tissues there. And what kind of more genes had a somatic mutation, for example here, ATM gene there, they create a tumor subtype B. Then some other genes create a tumor subtype C. So based on all those ideas and sort of cell lineage history there, you can start to understand the personalized cancer therapy, which basically we already tried it out in the Chinese patients. This is the prostate cancer in China. So it has been really tried all this surgery, chemotherapy, everything, and it really failed. So we sequenced this genome sort of single cell level, and we discovered this interesting pathway. They have a lot of copy number variations there. Then we found out drug actually could probably, potentially kill the disease there. And we discovered this drug has been used for kidney cancer, but not really on the prostate cancer. And then we use this drug, actually the patient is really under control. So it's more and more those patients right now in China, we started involved into this kind of trail analysis for the personalized cancer therapy. One of the important questions is that the genomics or omics? The answer is omics. It should not be just genotype there, right? You need to study the epigenetics, mRNA, protein level, metabolomics level, and so on. So you actually need to do more. And I'm not going to tell you more about that because of the interest of time. But I want to talk you one more sort of important factors, which are people tends to ignore, but it actually happens. It's very, very important factors. That's the environment. So even you know all the omics, they behave differently under the different pressure of the environment. So to really have your health in your hands, you need to understand the environment there. First of all, the virus, for example. In China, the liver cancer is really heavily caused by the HPV virus there. And this is one of the studies we've done, sequenced lots of liver cancer patients there, give you the sort of HPV integration of virus. This is the map up there. As you can see, it's non-random. So the different sites, the HPV integration were causing sort of different risk of having liver cancers there. Another one, which is not really virus, because the virus actually needs to be more discovered for the virome study there. We first initiated the project called sort of gut bacterial metagenomics analysis, just trying to sequence the bacteria in the guts. We first published this paper in Nature 3 years ago. So we've done something like 124 European samples that get 3 million genes there. And this is the latest development recently we've done. So we've sequenced over 1,000 samples from European, American sample, and Chinese sample. Then we actually get over 10 million genes in the integrated gene catalog. For those 10, consider how many genes in the human. It's only 25,000 genes there. But in your guts, you actually have 10 million genes, which are really, lots of them are metabolic related genes there. So then we started to understand, for example, if you look at, they have some of those private genes of individual specific genes, they reflect adaptation to the host immune systems. And sometimes they have common genes which are supply functions essentially for survival. For example, here, carbohydrates and amino acid transport and metabolisms there. Different countries are different, right? So we compare Chinese and Danish samples there. You figure out, for example, Chinese samples have more antibiotic resistance genes, probably reflect that we are using a lot of antibiotics there. And if you look at, again, for example, in the diets is a big issue there. They drink lots of wines. You actually discover that from gut. They drink lots of milk and yogurt too, so you can discover that from the guts too. But we actually have a totally different expansion of proteobacteria, which actually protect you to be very obese, which clearly is the case. If you look at amino acid, for example, the Chinese samples have more bacteria which are responsible for amino acid synthesizing and actually some of them vitamin synthesizing too. So again, that's the dice difference make us sort of lead this kind of bacteria to helping you synthesize the amino acids. And actually we have more carcinogen sort of carcinogen bacteria in your guts, which again probably we are exposed into this kind of food, which probably will have a problem. In a way, we were trained, but at the same time, it's not really nice. Just to tell you another examples about type diabetes, as I talked a lot about type diabetes there. If you look at 30 years ago in China, the type diabetes prevalence is only less than 1%. But now, recently, I think the latest number next year is 11%. This year is 11%. So we've already, another 14% are basically pre-diabetic. So 1/4 of adult Chinese is either diabetic or pre-diabetic. So it's really a lot of big issues there. It's hard to believe in the last 30 years you have dramatic changes in the genetic level. So it must be something different. It's the diet's difference or the environment difference or anything like that. So then we thought about gut bacteria. So then we sequenced the type diabetes patients' gut bacteria, we sequenced the normal bacteria, then we actually sort of compare them. We published this paper in Nature. It became very interesting, so we discovered many interesting bacteria there. Remember, all those odds ratios for genetics is only 1.4 or something. So here, we discovered bacteria which actually have odds ratio over 260. So they are really off scale. And those beneficial bacteria are really producing butyrate, which is really important category of bacteria there. They produce lots of butyrate producing bacteria there, which are really important for cell motility and butyrate biosynthesizing there. And if you look at type diabetes bacteria, there are many of those are oxidative stress resistance or drug resistance, which again, clearly some of them are really pathogenic bacteria there. And the gut environment is important. It's hard to just find one bacteria responsible type diabetes there. It's actually environment is very responsible there. As you can see, this is a very complicated network of different bacteria. How they sort of work together makes you a type diabetic or sort of healthy is a big issue there. And use all those bacteria again to have the ROC curve. It could easily reach over 80%. But for the human genetics, it's just random. Again, it tells you gut bacteria has much more effects on your health, or especially metabolic disorder, than the human genetics itself. We discovered 50 gene markers which you really could predict type diabetes well. So it's not just metabolic disorders. If you look at autoimmune disease, for example, the rheumatoid arthritis sort of patients we've sequenced clearly show the same thing. So you know exactly they have a link in the gut bacteria there. They have links for the cardiovascular disorders, for coronary heart disease, for example. You discover the gut bacteria has clearly association with there. Or cancer. So the colon cancer has been really proved already the gut bacteria play an important role. People, of course, are asking, is that just an association or is that actually a causal effects there? The answer is they are not just association, at least a lot. All these kind of bacteria are just association. So because of the diets and actually your host genetics have very important factors, bacteria there. So we started to try all kinds of diets to see how they change your gut bacteria. One of the diets we changed is the allele, for example, which is really, really good. We discovered that and sort of found it out this could protect you to be obese and diabetic. And if you look at all these bacteria there, are those short-chain fatty acid has been really boosted by this food, for example. Another way to do it is, can you find out the beneficial bacteria, then use feed it as probiotic to help you? The answer is yes. So we feed 3 groups of mice. The first one is high-fat food. The second one is high-fat food with this bacteria, probiotic. And this one is the normal chew. So after sort of 4 weeks of testing, even you are having high-fat food, if you have this probiotic, your sort of body weight glucose level is the same as the normal chew. So you have a very great potential to be probiotic for this bacteria, Riceberry bacteria. But anyway, so it just tells you the environment factors is always important. So with all these omics and environment, with 1 million people with self-reporting phenotype and very, very high quality data there with the whole genome sequence. So probably you will have a chance get your health in your hands. So this is about it. Thank you very much, and I have lots of people to acknowledge.
Great talk, Juan Rodriguez Cornell. So you were saying at the end there that you tried a bunch of different diets, and then you saw that aloe in the diet was protective for metabolic disease. Can you talk a little bit more about that?
Well, we just choose sort of aloe from this Chinese herb. You know, we have those traditional Chinese medicines herb book. So in that book, they say the aloe actually have a very strong effect against diabetic. So then we picked up that and started to feeding mouse. For example, we have different groups of mouse there. There's, you know, feed them aloe and normal diets, and aloe with diets and high-fat food diets and everything there. Then we discovered actually with very, very bad food like high-fat and high sugar content food or whatever. With this aloe, they will help you to sort of grow lots of those short-chain fatty acid bacteria, which will really help sort of protect you against the obesity and type diabetes there. So it become very interesting results there.
Yes, Walid Ridi from Saudi Arabia. I would like to know more about what is your opinion regarding why the mutations are clustered in that specific region?
Why the mutation is
clustering
in a specific region
on the genome?
That's a good question. Well, people think about it, for example, because of the DNA repair system and the replication systems there. When they tend to have mistakes, they have mistakes together, okay? This is one thing. And sometimes people think it's because of the GC contents is kind of different, or probably it's just highly repeated regions there. They have actually more possibility there. And nucleosome is another reason there. So they have lots of intrinsic reasons that maybe you will have those de novo mutations clustered together there. Yeah, and DNA coupled, transcriptional coupled DNA repair. That's another thing people were thinking. So there's lots of things, but not really has been firmed about which hypothesis is the right one. Probably all of them. Yeah.
Thank you very much, Dr. Jun Wang, if I'm correctly spelling.
Yeah, it's very nice.
Yeah. For the excellent presentation. I really enjoyed it. You gave us the future and your vision of genetic from a scientist's point of view. I'll be giving a wrap-up of this from the clinician point of view tomorrow in my presentation. And we see so many examples of what you said in our patients. For example, peptic ulcer patients where the gut bacteria is very important for the development of their peptic ulcer. Helicobacter and all these bacteria are very well known. And also we see other examples for other diseases. So it's very important to look to the environment plus the patient, as you mentioned. Now I have a question to you. You mentioned that de novo mutations increases with the increased paternal age in autistic patients. Now, we see this in so many of the autosomal dominant disorders like chondroplasia and others. Have you looked to such patients to see, this in them as well?
We should. We only look at autism right now, but we should expand it to other.
Because I think the spectrum will be very long.
Yeah,
not only for autism, but will be for other diseases.
Yeah, one of the important categories absolutely to look at the neurodegenerative disorders, because we know many of those genes are brain development genes there. So we know if we look at that, you will find interesting results. But we haven't collecting sample yet. So if you guys have interesting samples, it's good to it's good to collaborate.
Okay,
yeah.
Thank you.
One last question, please.
Thank you so much.
Okay.
I'm Dr. Muiz Bakhit. I'm professor of neurology and molecular medicine. This is exciting. I mean, you cannot have such excellent papers in Cell and Nature and Science, etc. But my question is related to this personalized approach for that cancer patient where you showed the AKT pathway is involved and you want to target that pathway for therapy. First, how did you come from the genomic sequence on that person to this pathway, and you define that pathway? And second, this pathway might be involved in the proliferation to be targeted for cancer therapy, or in apoptosis, because it may go through the caspase pathway or may go through the MAP kinase pathway and induce either cell proliferation or apoptosis. So how will you target that pathway?
So this is actually from an evolutionary perspective, because we sequenced many cells. So from those cells, you can have, as I just showed you from those one example of bladder cancers there. So you can infer the history of cell, the cell lineage history. Then you actually, from there, you can date all different kind of events of different kind of somatic mutation, different pathways into those events. So you know from there, and also some selection tools has been played here. So you know exactly what is the driver mutations there and what is the passengers in a way. So from basically you have different importance and different times of those mutations there. Then you can start it from there. But more importantly, actually, another, I didn't have time to say that. You have this xenograph, for example, on the mouse. So before you can actually doing it in the human individuals there, sometimes you can try out already in the mouse from those different drugs. You see if they works in the mouse model by the xenograph, you know, to get the real human tumors implanted into mouse. So then you know, then you can try back to the human. So we actually set up the same facility already in China in BGI to do that work. Yeah, thank you. So when I say the Million Genome, I try to explain it. It's not just a genome. It is a meaning omics plus environment. So as I just showed you this elephant idea there. So if you want to get an idea of any complex disorders, you need to see the whole picture. And the whole picture is not just DNA sequence. It is the entire picture of disease. Of course, people will think sometimes it's too ambitious, right? So why you are doing that? I mean, you cannot possibly collect a million data points with all this kind of omics data and environment factors there. But I can tell you, you will be there, no matter what. It's just a matter of time, but you will be there. And I don't know if BGI will be the first one to have that, but we hope we will.
Thank you.
Thank you.
Okay, thank you very much. Thanks a lot, Jun.
Thank you.